and provide innovation in this topic. Few words on the sugar consumption. As you know, the sugar consumption is increasing. This consumption is driven by many countries: India, European Union, China, Brazil, and US are increasing this consumption. And the sugar production is mainly due to the sugar cane, 75%, but also sugar beet is relevant in this kind of production. This crop, as I said, is so important in Europe. France, Germany, Italy are the leader countries in this sector. But the crop is damaged by many diseases, and we need a lot of research in order to be competitive on this sector. Before to come here, I select this slide because it's so interesting to show that in terms of calorie production, sugar beet is in third position after sugarcane potato. And this is a good example about the relevance of sugar for human food consumption. Few words on our main target at the University of Padova. The main disease, the main problem is a virus called rhizomania. The second problem is due to nematode, the beet cyst nematode, and the third disease is root rot. We are working very close with the seed company in order to develop improved varieties to able to grow and to produce in soils contaminated from these diseases. This is an interesting slide showing that the market share of varieties accumulated more resistance will increase. This is a good example showing that the double resistant variety will be grow until up 70% of the market share. This means that we have to add in current varieties more genes, more resistance in order to be competitive in terms of sugar production. Which is our challenge, to transfer the many traits from wild beet. C beet is the progenitor of sugar beet, and in the past we obtained many genes from this wild material in order to improve the disease resistance to sugar beet. In order to introgress this resistance, we have to continue the collection of these wild beets. This is a good example of a sea beet growing in Italy. From that plant, we obtained the first resistance to rhizomania used by all seed companies around the world. This is an example of the genes collected from that wild beet. We can see plants in a commercial field with the resistant gene against to rhizomania. This is a very aggressive virus, and plants without these genes coming from these wild species. This means that we have to continue the introgression of resistant traits, and we need the molecular markers in order to assist this introgression. In the past, the use of molecular marker was so slow. Now, thanks to the development of the SNP markers, thanks to the gene banks publication, we are really improving the capacity to introgress resistance traits from wild materials. We can see here that, for instance, that as you know, SNPs are increasing their popularity in general in crop breeding, but also in sugar beet, thanks to the development of the use of SNPs, we are really improving this process. An important innovation in the past was the release of the reference genomes. Thanks to this release, we update our knowledge, and at the moment we have a lot of SNPs and genes available in order to improve the sugar beet. Thanks to the cooperation with seed company, we develop a good protocol in order to continue the introgression of this resistance allele from wild materials. And this workflow is based on the three main steps: the development and a good protocol for automated DNA isolation, a protocol for high-throughput SNP discovery, and another protocol for high-throughput SNP genotyping. These three steps was the main demand coming from the seed company in order to solve their problems. We based the automated DNA isolation on interesting chemistry. In other words, we are, thanks to this chemistry, based on the use of magnetic beads, we are starting from a very limited quantity of leaf material. Normally we use 20 milligram of leaf material, a small leaf disc, in order to obtain 20 nanogram of DNA used, ready to use for the next step, SNP discovery, SNP high-throughput, SNP genotyping, etc. This is a method is really cheap, and we obtain good quality about DNA. This is a good example that a gel that I made before to come here, showing that from 20 milligram of leaf material is possible to obtain good DNA and without also quantification, because really we develop a good method that needs only the DNA isolation and the use of DNA directly without quantification for the next procedure. Thanks to the cooperation with Agrigenomics, a leader company from US working on RAD-seq, we developed in the past a panel of SNPs coming from resistant and susceptible individual lines. And thanks to this cooperation, we discovered a good marker linked to the key resistant traits in sugar beet. Now we are moving to use a very robust approach, the amplicon ion torrent sequencing platform, focused on the resequencing of specific regions known thanks to the recent release of a reference genome. For instance, this is an experiment design that we are testing now, including 50 resistant genes in a relevant chromosome of sugar beet in terms of presence of resistant genes. And we are at the moment analyzing data. We are quite satisfied about the results, because, as you know, we need robust marker in order to share this knowledge with seed company. And we found that this protocol is able to produce a robust marker without validation. We are able to introduce SNP identified thanks to the ampli-seq at the routinary level on the marker seed selection programs. About the high-throughput SNP genotyping, we are using a key instrument, the QuantStudio OpenArray systems, working on individual leaf sample. We are using at the routinary level this machine for screenings individual plants, and in order to remove, for instance, off-type plants, susceptible plants mixed with resistant plants. We can see here that this machine is so flexible, because we can use on the same machine 300 well block, and when we need high processivity, we can use an OpenArray block, in order to read a plate composed of 3072 well plate. In other words, when we need to increase our processivity, we move from the 384 plate to the 3072 well plate. It's a very key innovation for us. It's interesting to highlight that this plate is working with micro wells. In the sample is loaded thanks to the machine called AQFill. Inside this well, the well is hydrophilic and is able to retain the sample and to maintain the sample inside the well. Normally, as I wrote here, the volume is 33 nanoliters per well, a very reduced volume. We are moving from 384 well plate using 5 microliters per well to the 33 nanoliter per well. We reduce a lot the cost of TaqMan Genotyping master mix. This is the AccuField, this machine able to transfer the DNA plus the TaqMan Genotyping master mix inside a 384 plate. This machine is able to move the sample to the 3072 well plate. This is a very key innovation for us because we are screening more SNPs. For instance, we have a panel of composed of 256 SNPs for fingerprinting aims, and that we are using at the routinary level. This is a good slide showing that after the loading of the sample, the microplate is closed with a protected layer. We added fluid in order to avoid evaporation, and with the ready procedure, the microplate are ready to screen with the QuantStudio. We increased a lot our processivity because we are able to read four as I show here, four microplate every 3 hours, and it's clear that we really obtain a lot of data points. Each plate produce 3072 event amplification. In total, we have 12,000 data points per run. Here is an example of the curve obtained after the analysis of these microplates. And here we have a traditional cluster, allelic discrimination plots coming from the screening of this microplate containing many assays. Normally, as I said, we use 256 SNPs per plate. And as we can see here, the clusterization is quite good. This means that the TaqMan chemistry work very well with our DNAs isolated with automatic procedure. Another key innovation for us was the use of HRM on the same machine. Normally, we use the HRM in order to screen the sample to find some SNPs without to use labeled probes. No, as you know, the other approach needs to use the label probe, which RM, we can reduce the cost using normal PCR primers. And thanks to this pre-screening procedure, I think that the only machine available working with HRM on 384 plate is QuantStudio. This means that we normally analyzed 192 samples per plate, 2 replication per DNA in order to confirm data. And this method really allowed to us to, for instance, to reduce the sample sent to the facility for Sanger sequencing. Normally, thanks to this pre-screening, only 10% in our lab of sample contain robust NIPS ready to validate with the Sanger sequencing. It's a really good method for pre-screening and for SNP identification. Another very good procedure for us was the introduction of digital PCR. As you know, the individual genotype is quite time-consuming, and because we have to do individual DNA isolation. And sometimes there is a need from a seed company to bulk a sample. When, for instance, we are looking for a rare variant, a low frequency variant, we need to bulk to analyze the bulk of sample, and digital PCR is helping us to screen instead the individual sample. We are screening bulk of sample, and each bulk is composed from 100 samples. Now, at the moment, we are increase again the capacity of this machine, and we are working on a bulk composed of 160 individuals. This really improve our capacity of screening of plant in order to remove off-type plant. Like in this example, this is a greenhouse with many plants ready to screen, and working on that with this machine, we are able finally to remove, for instance, plants without the resistant genes. It's a very useful method to clean the seed lot. This is an interesting picture showing on the left the digital PCR well, containing 20,000 wells, and on the right we can see the chip after reading. We can see the fluorescent signal due to the amplification of the assay. Is a quite interesting highlight that the same assay working on QuantStudio for allelic discrimination is used also for digital PCR, and the analysis is quite robust. And in our experience, we reduce the capacity, the detection up to 1%. We really improve our capacity to detect off-type plants. For instance, we are able to remove on a bulk composed of 100 samples. We are able to remove heterozygote plants, for instance, coming from the contamination from other fields. This is the traditional allelic discrimination plot provided from the QuantStudio 3D. We can see the presence of the wild-type allele, and in this graph we can see here the presence of the negative allele. This means that we have to remove the bulk from the selection process. After the discussion of our workflow, I want to present our two case of studies. The first case study was so useful. We developed a method in order to assess the genetic diversity on our germplasm. This is a picture of our facility. You can see here that we are working with isolators. In each isolators contain parental lines ready to cross. And before the use of this method, it's clear that it was not possible to check plant by plant the hybridization process. Thanks to the introduction of this new workflow, we can check each isolator in order to verify if the crossing between parental lines is correct or not. We can see here an example of this fingerprinting based on the use of, in this case, 192 SNPs, showing that we obtain a good progeny coming from the cross of one parental and the other parental. In this graph, we can see the self-fed progeny. This means that we have to remove this isolator from this selection program, and this so is very useful because we can obtain progress only from the correct plants obtained thanks to the hybridization. Thanks to this innovation, we really clean, we discard a lot of material not obtained from the hybridization. It's so important to highlight that it's an easy procedure. We develop protocol able to screen a lot of plant, to fingerprint with a limited budget. In the past was not possible because other marker like SSR was not well adapted to this kind of projects. Another good example based on the use of this workflow is this study. We identify really good SNPs thanks to our protocol. we found, after a good screening, resistant genes coming from wild beet. I want to highlight the importance of wild germplasm in order to release a novel source of resistance. We found this resistant gene. This gene is quite impressive because we can see that the susceptible control showed in terms of nematode cysts per plant, a number of cysts per plant quite superior to the number of cysts per plant of resistant pollinators coming from the wild material. And we decide to develop, starting from that resistant line, thanks to bulk segregant analysis and SNP discovery, in order to develop the SNP marker and to use the SNP marker for the assisted introgression of this useful marker. This is a good example of the final results. We obtain a validated on all resistance commercial varieties a marker showing a complete association with the resistant gene, and we are using at the routine level, thanks to a good TaqMan assay, this marker in order to transfer the resistance and also to check if the resistance is present in the commercial field. We can see here that all 26 resistant varieties available on the marker are heterozygote and contain the new discovered genes. Because it's a very important highlight that is good to find good marker, but it's also important to find good assay in order to screen this marker on the commercial production of seeds. We are continuing the history. We are developing our other marker for other key traits like bolting resistance, and these achievements are strictly due to the workflow used. We really hope in the future to continue this progress using the AmpliSeq, because we really increase our capacity to produce useful SNPs, and we really hope to be here next year with other results and to provide these results with other breeders. In summary, our workflow increased our capacity and the speed to improve the breeding programs actuated by the seed company. And this achievement is always due to cooperation with other scientists. We are cooperating very closely with the USDA in terms of exchange of materials, and we hope to continue also in the future this, to have benefit from this cooperation with other countries. This is our campus at the University of Padova. We have a good genomic facility in order to carry out this project. And I want to conclude this with this slogan: we are moving from the nucleotide to the field. Thanks for the attention. The main benefit is the yield advancement, because it's clear that after 4 years we are able to estimate the advancement in terms of yield benefits, because as you know, the seed company are looking for improved varieties, and we are really improving this process by releasing new varieties with a better yield, and yield is due not progress on yield potential, but progress in terms of real yield, resistance to diseases. We understand, thanks to this cooperation, that our yield improvement is mainly due to the introgression of resistant genes. Thanks.
Thank you very much.
Thanks.
Can we do one more question?
One more question?
Sorry. Really quickly, thank you for the interesting talk. I was just wondering, on your QuantStudio platform,
Yes.
You're handling per machine and how many genotypes on each sample, how many targets you're
Yes. Yes, thanks for your question. It's clear that working very close with seed company, we have to very calculate very well, no, the processivity, the cost, and it's clear that we are improving our capacity. And we are calculating every time the new technology in order to improve, no, to again our processivity, and the cost, to be honest, are updated day by day because, for instance, thanks of the introduction of digital PCR, for instance, we really reduce the cost because, as you can think, working on bulk of 100 leaf sample, instead we reduce the cost of DNA isolation very significantly. This means that the cost calculation is well updated day by day, and we are always looking for the new technology in order to reduce again the cost. But I want to stress of the importance of, for instance, of digital PCR, because for us the major cost was the DNA isolation, and now thanks to the digital PCR, we really improve our screening capacity, and at the same time we reduce the cost of the analysis.